Aha! Sweet with a tang. You must have eaten a Malta. A Malta?
Malta, a cross between an orange and a sweet lime, a hybrid with the goodness of both.
Dolly. What does the name Dolly mean to you?
Dolly, the most talked about sheep ever, cloned from a single cell of an adult sheep's mammary gland.
And there's no way you have not heard of insulin.
Insulin, the hormone that regulates energy and glucose metabolism in the body. For diabetics, it's a lifesaver.
Welcome to the world of biotechnology, the world of genes and clones, crossbreeds and trait selection, the world of recombination and creation, the world of scissors and glue.
Welcome to the Biotechnological Revolution Gallery of the VITM Bangalore. Biotechnology has been around for centuries. Fermentation, seed selection, crossbreeding of animals and plants. Man has been using life in various forms to his advantage. Having learnt about the DNA and genes and how they worked, man developed a modern super speciality. He could now literally cut and paste genes, recombine them. He could engineer traits by modifying genes in specific ways to biosynthesize desired products. Knowledge of enzymes and DNA gave man scissors and glue, the tools to engineer life. For biotechnology is just that. It uses cells and microbes as factories and enzymes as workers.
With a pair of scissors and glue, it is so easy to create a new design. What if we could do this at the level of genes? That is what genetic engineering is all about, and we will explore that today.
Amazing, but true. Wherever we go into this world, whether plant, animal, bug or microbe, if it is alive, it will use the same language and follow the same dictionary of life. The dictionary of ATCG, where A is adenine, T thymine, C cytosine and G guanine. At the level of cells, all life is one. Once the genius of Francis Crick, James Watson and Rosalind Franklin had discovered the secret of life, the structure of the DNA and its mechanism, it did not take long to unravel this dictionary of life. The genetic code was cracked, and scientists soon discovered that whether in a bacteria or a human cell, one specific code stood for one specific protein. This knowledge became the backbone of genetic engineering.
Let's start this exploration with the genome. The entire code of life packed in the DNA. The genome of every single individual is unique.
The Human Genome Sequencing Project started in the year 1990. Its goal: to understand the genetic makeup of the human species, map the genes, break the code. Interesting but true that the DNA of chimps and humans is 98% identical. Among humans, the difference is only 0.2%. So how did we go about reading this book of life? The DNA is taken from the human chromosome and shredded into short segments. An automated machine then reads these sequences. Remember, there are just 4 letters in this dictionary. The next step is assembly. The computer reads the segments, finds overlaps and matches them. It looks for letters that signal the start and end of a gene. Annotation is the third stage. Now, with the full sequence in place, one can find genes that protect or cause disease. The Genome Project has enabled scientists to read the DNA like a book, separate the nonsense from sense. 3 billion base pairs, more than 40,000 genes. Genes that code for protein synthesis and control all functions of life. Along with the human genome, the genomes of yeast, E. coli, fruit fly, worm and mouse were also cracked.
Fly, mouse, yeast, man, diverse organisms, but similar genome. This knowledge became integral to bioengineering. But man needed something to cut the genes. He needed scissors.
Could scientists cut out a specific gene and introduce a new one? Could they engineer traits? They needed tools, molecular scissors. Imagine discovering just that, something that identified a particular sequence and cut it. Restriction enzymes. Their discovery was a revolution. Isolated from bacteria, they could be used to cut strands of DNA into defined fragments. Restriction enzymes became the backbone of genetic engineering. They could identify sequences and snip them at precise locations. At the gallery is a working model. Watch how, at the press of a button, the DNA is cut at a particular recognition site. One learns how each restriction enzyme recognizes a short, specific sequence.
Tools to cut and isolate. But man needed something to put things together. He needed glue.
Ligase, the glue, something that can actually bind molecules, glue them together. Ligase are specialized enzymes. Like all enzymes, they are proteins and highly specific in shape and task. When scientists found DNA ligase, they were thrilled. They had found the specific enzyme that made DNA repair, DNA replication, and even DNA recombination possible. DNA ligase was the glue that could help scientists glue genes together. Gene splicing was born. The gallery shows how. Gene splicing is a process of cutting and rejoining genes on a DNA molecule. The DNA after splicing is called the recombinant DNA, and this is genetic engineering. Let's see how it works. The restriction enzyme, the scissor, cuts the gene of choice. This isolated gene, the code for the desired trait, is pasted on the DNA with the help of a DNA ligase. This is the recombinant DNA. This engineered DNA can be introduced in bacteria that act as factories. Each new copy carries the desired product.
But aren't we missing something? We have scissors and we have glue. But how does one get to move the gene of choice from one place to another? How?
Bacteria are amazing. They are prokaryotic cells. Simple, primitive. A naked DNA and enzymes floating in their cytoplasm. But bacteria have something unique, something more. They have plasmids. Plasmids are extra rings of DNA. Scientists soon discovered that this extra DNA in bacteria, the plasmids, could be important tools for genetic engineering. They became the vectors. Let's see how. That's the plasmid in E. coli. It is cut with a restriction enzyme. The gene of choice is added and glued with the help of DNA ligase. This is the vector, the modified plasmid. This genetically modified plasmid is added to a bacterial colony and allowed to grow in a culture. The rapid rate of growth of bacterial cells make them ideal factories. In the culture, all the new bacteria start expressing the inserted foreign gene. Hormones, enzymes, medicine, pesticides, even new strains of crops and organisms. The applications of recombination are many. At the gallery, one explores the applications in the field of agriculture and livestock. Agricultural biotechnology is a precise scientific process used to improve plants, animals, or microorganisms for the benefit of mankind. It allows scientists to add a single gene from a donor to improve the commercial plant variety. What does this mean? Imagine removing the gene that makes tomatoes ripen very fast. The modified tomatoes would then remain firm and fresh for a long time. Onions make us cry. Their cells have a volatile sulfur compound that irritates the tear gland. Biotechnology can actually remove the sting of an onion without changing its flavor or nutrients. All that needs to be done is remove the gene that produces the eye irritant. Cotton, a cash crop for India, often attacked by the bollworm. The moth lays eggs on the leaves. When larvae emerge, they feed voraciously and harm the plant. Bt cotton tackles this pest. Bt cotton has an added gene for the production of an enzyme isolated from the soil bacteria Bacillus thuringiensis. This enzyme disrupts the inner lining of the larval cut. The larvae feed on the plant and drop off within a few hours. Larvae gone, the plant grows well.
What holds good for plants holds good for animals too. After all life is one at the cellular level.
Biotechnology and genetic engineering have touched all aspects of life. By recombining genes and therefore doctrine traits, one can get cows that produce more milk, or sheep that have more meat or fur. Crops that fight disease and pest, that need less water or manure, sweeter fruit, abundant seed. At the gallery are displayed normal products alongside genetically modified ones. The difference is obvious.
The difference is indeed startling. And sometimes it's the difference between life and death.
This woman has diabetes. She's just one of the more than 3 crore people in India living with this condition. Diabetes is a condition where pancreas do not produce enough insulin. Insulin is vital for life. It maintains the level of sugar in the blood stream. In the 1920s, patients of diabetes had a 3-year life expectancy after diagnosis. It was a killer. 1923, insulin was discovered. Life expectancy increased to 30 years. The source of insulin was the pancreas of slaughtered pigs and cattle. But genetic engineering made the production of human insulin possible. Today, large quantities of human insulin is produced world over. The gene that codes for human insulin has been introduced into bacteria. The bacteria produce the human hormone. Today, patients can not just monitor their blood levels, but even take shots of insulin themselves. They can lead normal, healthy lives. Biotechnology has aided man's war against cancer. Tissue banks, research facilities, gene experts, scientists are studying cells and figuring out the causes and treatment. Cells grow and die in a strictly controlled way, controlled by the DNA. There are genes that switch off and on. Sometimes they malfunction. The result, an uncontrolled division of cells. Cells grow continuously and take over, lump to form tumors. If these cells are malignant and produce toxins, they take over healthy cells. They invade the body, wreak havoc. Scientists have isolated genes that cause cancer. Anti-cancer drugs are being developed. Cancer is now curable. And who knows, maybe someday we will have the technology to switch off the genes that cause cancer. The products of genetic engineering have been life-saving for many. Growth of the human body is regulated by a human growth hormone. Deficiency or overproduction can cause severe problems. The human growth hormone has been produced inside bacterial cells using recombinant technology. Our lungs and other internal organs are protected from damage by a protein, AAT. People with AAT deficiency suffer from lung damage. AAT has also been produced in living factories and protected the lungs of many deficient humans.
Blood or spit, urine or a single strand of hair. Each cell of our body has a distinct signature, a fingerprint. Bad news for criminals.
Physical evidence like fingerprints, yes, but biotechnology has revolutionized crime detection too. The police now looks not just for the physical marks, but cells of the body of the criminal. For inside each cell lies the fingerprint. DNA fingerprinting is here to stay. An interactive display takes the viewer on a step-by-step exploration. The DNA is isolated from the cells collected from the scene of crime. The second step, the DNA is cut into pieces. One gets fragments of varied sizes. The third step, using gel electrophoresis, the fragments are arranged in order of size. One gets a unique pattern this way. This is the DNA fingerprint, and this never lies. Gel electrophoresis. Let's play and understand. Tilt the board and watch the movement of the balls. Each ball has a different size. It's quite clear that the movement of the balls inside the gel-filled box depends on its size. The bigger balls move slowly as they experience more resistance. They lag behind. The balls occupy different positions at different points in time. In biotechnology labs, gel electrophoresis is a handy process. The DNA cut into fragments with restriction enzymes and placed in a gel. A charge is applied and the fragments move, creating a unique signature. This way, they are identified and picked up.
How can we understand biotechnology without getting to know Dolly, the clone? So, what is cloning?
Dolly, world's most famous sheep. Sheep, like almost all animals and plants, produce sexually. The male and the female unite to create an offspring. But Dolly was extraordinary. There were no gametes, no fertilization. She was created from a single mammary cell. The cell nucleus from this cell of one sheep was removed and transferred into the unfertilized oocyte of the surrogate sheep. An electric shock stimulated cell division and Dolly was cloned. Cloning proved that a specific part of a body could grow into